like the last 3 months.
All right. Well, let's go ahead and kind of dive back in here and take care of part 2 of this, which I think will have a lot more. There's a lot more medical conditions, a lot more questions you might come up with about genetics and in general medicine in relationship to it. So we talked about gene therapy is changing disease prevention and treatment. Likely in the future, more and more genetic targeted treatments will occur, and in your practice as a healthcare provider, you will be exposed. You'll be seeing a lot more patients being treated with these sorts of things. Okay. Number 11 here, summarize the medical impact of low-cost, high-speed genomic sequencing. So what might surprise you is the cost of sequencing has been dropping faster, and the speed at which it's happening has been going at a faster speed than Moore's Law. You probably don't even know what Moore's Law is. Maybe you do. Anybody knows of Moore's Law? Ever heard of Moore's Law before? It was I think it was Frank Moore back in the 70s or so. He made a prediction, and he said it seems like processing speed roughly doubles, and the cost per transistor roughly halves every 18 months in the world of computing, in creating computer chips. It was back in the 70s, right? Giant computers, very slow, couldn't hardly do anything. Well, it's pretty much held up to that every 18 months, processors are getting twice as fast, and they're also about half the amount of cost per transistor. It has radically changed our lives. I mean, I'll have to admit, maybe we're so surrounded, we don't even recognize it, but I'm holding a little wireless device here that magically somehow talks to the screen up here, and whenever I write, something happens. It also records my voice, and cars can start to drive us around, and there's all kinds of machines doing incredible things, and that's largely driven by increasing computing power available to drive those processes. So why do I mention all of that about genetic sequencing? I mention it because genetic sequencing, I'll just give you highlights. 2001, when the first sequence was ever sequenced, it was a composite sequence from over 100 individuals. The Human Genome Project, 2001, cost the government over $3 billion. Took years and years and years, over a decade to get that done. No more full sequences of humans sequenced until 2007, and that was James Watson and Craig Ventnor. James Watson, of course, being co-discoverer of the structure of DNA, and Craig Ventnor was one of the pioneers of DNA sequencing. Their genomes in 2007 cost about $10 million and took several months to sequence. 2010, 11, getting closer now, to 2010 or 11, sequencing costs were about down to $10,000. It only took about a week to get a human genome. Okay, 2014, human genome finally dips below the cost of $1,000. You can sequence it in a couple of days. Where are we at today? We are well below $1,000 per human genome, and it takes less than a day. Now, what does that mean? It means that you can get all of someone's sequence in a very short period of time and very low cost, and that means there's just wealth of information that is just like a tsunami of medical information that's rolling through medicine right now because of that change in technology. And we'll get more into that, I think, in some of the diseases, and I'll try to explain more, but you're welcome to ask questions. I'll show you towards the end. I have 23andMe pulled up here. I'm not going to recommend this place in terms of, I think you should do it, but I will show you how it works. Ellen, myself, and the kids have done it, and so I can show you what it looks like. It's impressive of the information you can pull out of that. Not always perfectly accurate, but it's fairly impressive. All right. Questions? So that actually leads to the last point. With all this information, what are you going to do with it? Well, on the medical side, that's why genetic counselors are so important. Genetic counselors are incredibly important, and we definitely need them. These are individuals professionally trained in both psychology and in genetics to be able to walk patients through understanding their own genetic information and then the implications it might make on the decisions that they have in front of them. Examples. Cancer is the big one. Suppose someone has colon cancer and they go in And now part of standard treatment for colon cancer, you get screened to see if you have genetic mutations that made it likely that you had colon cancer. If you do, then now you've got some information to wrestle with. Number one, that actually informs you about your relatives. Your relatives might also have that same gene. They might be at high risk, right? And so there'll be a decision on, well, should I share this information with my relatives or should I not? And the genetic counselor is there to help kind of walk through those sort of decisions. If you have kids or are thinking about having kids or not having kids, it impacts that decision. Prophylactic care. Many of you have heard about of Angelina Jolie, right? Prophylactic bilateral mastectomies because of having genetic testing indicating that the BRCA1 and BRCA2, she had mutations within that, dramatically increasing the risk for breast cancer and ovarian cancer. Questions? Yes.
The reason I crossed out that outcome there is because the specific information off the BRCA1 to gene mutations, it was briefly mentioned in the physical assessment, and will all be talked about again next semester.
That's fair. That's helpful to know. So they'll not be tested over on this unit, but it's other parts of their curriculum.
But it is a good example of...
Yeah, it's just a nice little example there, yeah. And the nice thing is nurses can get trained as genetic counselors. I don't know if you knew that or not. So by and far and away, the most majority of genetic counselors are they're trained with a master's degree after their bachelor's. That's the most common way. You can apply to these programs and get in and go on from there. But there also is a track in nursing master's level. Get a master's in nursing certified as a nurse genetic counselor to help, again, patients understand their own genetic information and be able to walk them through decisions. Good. Any questions on anything we see here? Okay, well then let's switch over to the part 2 of this unit with genetics, and I'll put that up there. Yep, perfect. And this talks about genetic and congenital disorders. That's almost a misnomer. Man, that's basically, congenital just means the defect is present at birth, or, the problem, the fundamental cause of the disease was present from birth. And so really, these are almost interchangeable in a lot of ways. So if you see those two terms, all genetic diseases are congenital. Now, I guess there technically are a few, perhaps, congenital diseases where environmental things happened in utero that caused the disease, not genetic, but environmental. That's still congenital, but nonetheless, they're very, very related. Oh, it actually defines it all right here. I guess I should have just looked down. Save me some thinking power there. Yeah. Yep, that's all true. Okay. I don't know if there's anything else you want to know. I'm not sure what to say about that, but it is, yep. The only thing I didn't say was that, causes of congenital disorders could be defective genes, could be chromosomal abnormalities. I didn't mention that. And then, absolutely, environmental factors also can do that. Now, it is true some genetic disorders may present at birth, or they can appear way later in life, right? Huntington's disease or something like that, where perfectly normal, everything's going great, and then you get a very terrible disease later in life, very much genetic, very much was kind of predetermined in the beginning, but was not evident until later in life. Okay. Okay, so here we go. Here's our first category of diseases. I think we'll do several of these right in a row here. Autosomal, dominant, inheritance. So we know the autosomals, the 1 to 22 chromosomes. We know the dominant. What does that mean again? If they have at least one copy of the bad gene, they're going to have the disease. And inheritance just means increased inheritance. Here's the picture of a. Oh goodness, I'm blanking. This is how bad I am, Ellen. Whenever I don't teach for a long time, I can't think of all the words. It's a pedigree. Here's a standard pedigree for an autosomal dominant condition. The colored boxes are individuals who are affected. Circles are females, squares are males, and then the little crosshairs means that there was children from that relationship. And then, so for example, this top family up here, there are 4 children. So 1, 2, goodness, get some numbers there. 2, 3, 4. And children 1 and child number 2 are affected. Child 3 and child 4 are unaffected. And then if you look down here, child 1 had a relationship with this male, and they had 4 children of their own, and sure enough, 2 of their 4 children were affected. Okay? So the question asks us, or the learning objective says, what's the percentage of children affected or not affected in these two conditions? What's the answer to that? It's 50%. 50%. So let's imagine, let's do the little Punnett square. Remember that Punnett square? Let's say this parent up here they have the disease causing one, the capital D for the dominant allele, and then they have a little d for they have maybe one normal recessive copy, and then their partner has a normal allele. He's homozygous for the healthy type, and then that couple is having children. Take a look down here. If I put like this, and like this, and then over here, like this, and like this, and I remember that all of my categories always end up having to be 25%. Take a look here. This and this, that's 25%. This and this is 25%. That. You see how I'm filling this in, right? And then the 2 little ones. Okay, so each of these are 25%. So right here, this one, these kids would be unaffected, right? That's 25%. Plus, these would be unaffected, right? That's another 25%. So 50% are unaffected. And then look at these other 2 over here, though. If they have at least one copy of the capital D, then one copy of the dominant allele, get at least one of those, you're going to have the disease. That's 25%, that's 25%. So that's where your other 50% comes from. So 50%, and then if we actually just look at the pedigree, from 2 of these 4 kids have it. Look further down, 2 of those 4 kids have it. Now with small numbers, it doesn't always work out that way, right? You could have 3 kids, none of them affected. You could have 3 kids, all of them affected. It's kind of the roll of the dice each time, but on average, the dice or the thing it's a coin. It's a 50-50 chance, right? Every child is born. It's just you're flipping the coin. Will this child have it? Will they not? Will this child have it? Will they not? All the way down. Okay, questions on that? Be able to list examples. That's also what it says too. Examples of disorders. Here we go. These are all ones that you should be, at least familiar with that fall into this category. Adult polycystic kidney disease. I don't know if you've heard about this one or not. Probably not. You had it last semester?
Next.
Oh, they have it next semester. Okay. Polycystic, this just means mini cysts. And in the kidneys, if you actually look at the kidneys, I remember dissecting a cadaver over in the science and math building. One year we were pulling the new body in, and sure enough, this individual had polycystic kidney disease, and the kidneys were just filled with cysts. So it was just like the cysts were all about an inch in size, and it was just all over the kidney. So if you just Google it, it's kind of a little gross, but it's not that bad. The kidney fills up with cysts. It's a genetic condition and dominant. Huntington's disease, probably this is the biggest one that you might want to spend some time focusing on. This is that terrible disease where it is progressive loss of coordinated motor function, also progressive loss of mental function leading to dementia, usually resulting in a shortening of life, and it is dominant. This is that really tough one where this is probably the one key disease that everybody talks about that they're not sure if they would want to know or not because how much it messes up your life. Like, oh goodness, I remember listening to it was an interview on the news with a young lady. She was in her early 20s. Her father had died of Huntington's disease, and so she just decided to get tested to find out. And of course, she found out that she did have it, and so she wasn't sure when it would affect her. Her father had died in his 40s, but it's kind of like a ticking clock, right? Just kind of a ticking clock above you, so you're kind of wondering, what career should I choose? Should I get married? Should I have kids? What should I do with my life? Because now it's very much different than maybe what it wouldn't be. And some people are like, well, I want to know so I can plan that. And other people are like, no, then my whole life I'm constantly have a rain cloud following me about thinking about tomorrow. And so it's really tough. These are tough things to think about, that's for sure. And no good treatment, unfortunately, for Huntington's disease. Be aware, since this is autosomal, I didn't mention it when I was going through, but what do you think the percentage of males versus females affected? Is it equal? Is it different between males and females if it's an autosomal disease, autosomal genetic disease? It'd be equal. That's exactly right. It has nothing to do with the X chromosome, has nothing to do with the Y chromosome, so it's going to be equal for everybody. Right? This is a very egalitarian sort of disease. Doesn't matter if you're male or female, same percentages. That's why we see on here there's some males affected, there's some females affected. There you go. Okay. And then you can just kind of read through the rest. I think your book has brief descriptions on most of those there. Now let's talk about a specific example. This is Brugada disease. And this is a pretty terrible disease. It used to be called, what was it, sleeping death? Oh goodness, it was something along those lines. The reason why they called it that is because individuals with this disease will often just go to sleep at night, everything's great, they're perfectly healthy, and they don't wake up. Why? Well, they have genetic mutations with their hearts that make them at a high risk to have electrical problems that result in sudden cardiac arrest, right? Just kind of out of the blue. Now, it runs in families, though, so they can kind of figure out from their inheritance of, like, here we go, a young man's father died of this syndrome, sudden death, there's the gene mutation, it's dominant, mother and grandmothers are alive and healthy. What's the son's chances of having the disease? So, what do you guys think here? What's this point about the mother and grandmother being alive? Why does that matter? Yeah, kind of X-linked. It's also just kind of confirming that, so Brugada is autosomal. It's also confirming that they're still alive and that he's not the unluckiest person in the world and like he's got this on both sides of his family. That's kind of like the main thing here is we're saying, okay, dad obviously probably had this, but mom seems to be normal. So we have a genotype of dad probably had at least one bad copy of it. What's the son's chances of having the disease? 50%. Yeah, autosomal does 50% chance. I don't think this is in your learning objective, so I may not do too much on this, and don't take this too far. But penetrance is just a fancy way of saying, unfortunately, things aren't quite as simple as I wish they were for you. And it turns out some diseases, even though they're like autosomal dominant, they won't be exactly 50%. Well, why not? Well, some diseases actually have something called penetrance, and what this means is, even if you have the trait, it's still not a guarantee you'll actually have the phenotype in the disease. Even if you have the genetics for it, you may not guarantee get the actual disease. And things with high penetrance are kind of like automatic, right? There's really high penetrance, 100% penetrance. If you have the DNA, you have the disease. Low penetrance, that means even if you have the DNA, you might not get the disease. Right? Because there's a decent chance that you could come out okay. That's all that means if you see that term penetrance used. All right, here's our second one, autosomal again, but this one's recessive inheritance. Describe the percentage of children who are affected, not affected, and those who become carriers in an autosomal recessive. So we see here in this picture the individuals that have the halves, the dark half, that means they're carrying, like for example, both the parents up here, they're both carrying a disease allele, but they also have one normal allele. And then for the kids, this child on the far left has two bad alleles, so therefore has the disease. And then, so that's child 1. Child 2 is heterozygous, so is a carrier. Child 3 is has 2 normal copies, so not even carrying the disease. And then there's 4, there's another one that's carrying the disease. So question for you, help me work this out. What's the percent of children affected, not affected, and those who become carriers? Let's start with affected. What's the percent of kids we're going to expect, on average, that'll be affected by the disease? Yep, remember that we did that in the last lecture? We did that whole math, everything. We added it up. Those are the ones that have to inherit 2 recessive alleles, and so that chances go down, so it's 25% are the ones for the affected. What about the not affected? The not affected ones? 25%. Yep, same thing there again, actually 25%, because they're the ones with the normal dominant alleles. Those are the normal in this situation. And then lastly, of course, what's the carriers? 50. Yeah, carriers for, there'll be lots of ones that have the heterozygous phenotype. Okay? There's actually a lot more diseases that are recessive, and it makes sense there's more recessive diseases, doesn't it? Just think about this. If there's a bad dominant disease, it burns itself out, right? It kills everybody off. It can't last for very long because, like for example, with Huntington's disease, it's terrible. I mean, it really is a terrible disease. It actually not only has is an autosomal dominant, but it actually has what's it called? Anticipation. Anticipation. It's a fancy term that basically means every generation it occurs sooner in that individual's life. So it'll start with someone's grandfather who's 60 years old who develops Huntington's disease, and then it'll affect his son or daughter when they're in their 40s. It'll affect their child when they're in their 20s, and if it gets to the next generation, it'll affect the next generation when they're in their 5 to 10 years old. Well, they can't reproduce, so that's the end, right? That kind of burn. So there's actually a lot more recessive diseases because there's a lot more of us that can run around that are healthy that have these recessive diseases that it's not affecting us, and we can reproduce and have. Sorry, I'm talking so blunt terms here. We're walking around reproducing. Go ahead.
They're out of the kids, there's 25%
Yep.
chance that one will be affected.
Right.
One is affected. Looks like he's married to someone who's not affected.
Yeah, talking about number 1.
Number 1.
Yeah, so she's affected.
She's affected. Someone who's not affected, but
Yep.
all of their children have the recessive.
Yeah, isn't that amazing? Yep, because if we did the Punnett square on that, that's how it works out. That's exactly, so it's not, so these numbers apply to, in general, like the big picture kind of thing.
Got it.
Or even more specifically, they apply to this pairing right here.
Okay.
Yeah. Yeah. That's good question. Very good question. Any other questions there? So cystic fibrosis is most common autosomal recessive disease. Boom, cystic fibrosis. You got to know that one. So make sure you file that one away. There's all kinds of glycogen storage diseases like Tay-Sachs. You may have heard of Tay-Sachs disease. That's a glycogen storage disease that's recessive. This oculocutaneous albinism, PKU. That's one they screen for at birth. For all kids everywhere in this country. A little stick of blood from the heel, right? When the baby's born, they always do the standard genetic testing. Depends on the state, between 20 and 60 different diseases that they'll test for. That's one of them, actually, because if you can figure that out early, you can change their diet so they don't end up with consequences from having that particular DNA difference. Sickle cell, and then, there's Tay-Sachs. Ironically, Tay-Sachs is a glycogen storage disease, but fair enough. Questions? Any on that? That makes sense, right? Autosomal recessive inheritance.
I want to mention that there's a nice table at the beginning of your chapter that talks about some examples.
Nice. I think that would be helpful just to review that, make sure I'm up. And so if I saw a test question that gave me a particular disease, I'd be able to connect it in general with the one that, what category am I in, right? Is this an autosomal recessive or whatever? As well as being able to know what's my inheritance pattern with a particular disease. If it's autosomal dominant, expecting 50% of the progeny to be affected or not affected. Okay, so question, following statements true about an individual who's a carrier for cystic fibrosis gene. So, if they're homozygous, suffers from cystic fibrosis, okay. Homozygous does not suffer from cystic fibrosis. Heterozygous suffers from cystic fibrosis and heterozygous does not suffer from heterozygous. Which of the following statements is true about an individual who's a carrier? What do you guys think? Remember the definition of a carrier? A carrier is someone who's heterozygous. They've got one good copy, one bad copy, and they're carrying. What I always remember is I just think about them. They're carrying around one bad copy, but nobody can see it. Right? They're literally wherever they go, they're carrying this one bad copy. They look perfectly normal, but they're carrying this one bad copy, and that means that if there's two carriers, they can potentially have a child who's affected. Only in recessive diseases. Okay. All right, so then now we have the X-linked or sex-linked traits. So on these, it's almost always talking about the X chromosome. And actually, I think I have a slide that actually shows. Yeah, here we go. So here's the deal on this one. They actually even show it in kind of a different format. But we have an X-linked condition here. And this particular individual at the top, this is a female who is a carrier. And females can carry X-linked diseases, but males cannot be carriers of X-linked diseases. Why? They literally only have one X chromosome, so they can't have an extra X chromosome to compensate if they get a bad copy. If they get one bad copy, they have it, right? So in this situation, this individual has one good copy of the X chromosome, whatever trait there is, and then maybe one recessive bad copy there. This partner looks like, of course, he has one Y chromosome, but he's also got one normal chromosome. Look at the children here. Again, we'll do 1, 2, 3, and 4. What percent overall of the kids are picking up the, well, I'll be very specific here. What percent are at least inheriting the genetic recessive trait? That's probably a little bit unclear. I can see why you're cautious on that. Let's be even more clear. What percent are likely to be affected? And we actually have to break this down by male and female, right? It's going to be different between males and females. We have a sex-linked one. So let's do specifically males that come from a pairing that looks just like this. What percent of males would be affected? Yeah, it's actually 50%. It's 50%. And here's fundamentally why. If you look at the mother here, she has one normal and then one X chromosome that's carrying the disease. Whenever any egg is created from that female, each egg is going to have only one X chromosome in it. It's either going to be the diseased one or the regular one. And if the male progeny won't have any other X chromosomes, so it's a 50-50 chance on whether or not the egg will originally have the healthy X chromosome or the affected X chromosome. And you can really just trace it back to the maternal inheritance. Now, if you talk about females in this, what's the chances that they will be affected, that they'll actually have the disease? It's actually 0 chance in this particular pairing because they're going to always get a normal X chromosome from dad every single time. And this, in this particular pairing we have at the top here. The only way, it does happen, the only way that a female can be affected is if dad is actually carrying his X chromosome, if he's actually affected. Does that make sense? In fact, let's see, I'm doing all this work, I could actually look below. Do they have a female that has, no, they don't even put it's so rare, very rarely happens, but a female would literally have to have both the father and the mother passing on the X chromosomes. Does that make sense? So this X chromosome, I should switch it to the little x. What's that? What's?
That's a little typo there.
Where's the mid? Oh my goodness! Hey, wow, look at that! Woo! I tell you what, that's interesting. That we're not getting into that today. I'm sorry, that is not on the agenda. Not tackling that one. I'm sorry.
I was wondering.
There we go. Fixed. There you go. Okay, that was confusing. So does that make sense? So 50% of male offspring will be affected because of the maternal inheritance. Very, very infrequently will females ever be affected, and to have them be affected, both parents have to have been carriers or actually have the disease in the terms of the father. Okay? Here's the examples you should be aware of. One of them is, for example, Fragile X syndrome. This is on the X chromosome, and again, we basically have, in this situation, here's on the left-hand side a normal X chromosome. On the right-hand side, that's the Fragile X. There's actually this, they even list it, this triplet repeat where it repeats the same nucleotide again. Often it's like CAG, and when the DNA is being copied, the polymerase that's copying it gets confused whenever it's going across a long section of repeats, and sometimes they'll add too many repeats in there, and when it does that, it can mess this part of the chromosome up, and you can actually see it underneath a microscope here, see what it causes. Okay. In this particular disease, the main outcome, unfortunately, is intellectual disability. Okay. I think we kind of covered this one here, so I'll let you maybe go over that. The answer is 50% son.
Would you be able to go back to that Fragile X?
Oh, yeah, absolutely. I'm sorry if you were trying to capture all that. Yeah, I don't... Okay. Let me see here. Where was it?
Post this version of the...
You'll post this version to the whole thing? Okay, good. I'm not going to go over further in depth on that. The main thing is note that Fragile X is an X-linked, so you can categorize that. And number two, I think at least be aware of the major outcome, which is intellectual disability that comes out of that. And of course, again, it's going to be affecting males more often than females based on the genetics. Okay. See if I can get this to go forward here. All right. So, quick discussion question. If you have the allele for a trait, do you display the trait? Yeah, quite literally, the answers are down here below. It very much depends on what kind of trait is it, right? If it's the recessive and you only have one copy of it and you have the dominant, nope, you're not going to see that trait, right? If it's a dominant one and you only have one of them, yeah, you're going to see that trait. And then the sometimes is the intermediate penetrance, and again, that's that whole thing about even if you have the DNA for something, it's still not always guaranteed with certain diseases that you'll actually express the trait due to other factors. Okay. Genetic imprinting. Wow, I don't think we cover that. Yeah, I mean, this is awesome stuff, but I don't think that's not the...
I left it in case you wanted to...
Oh, in case I want to talk about it. Wasn't that nice? That was very nice. We'll see if we have time. If we have time, I might come back to it. Just want to make sure I cover all of the learning objectives. Did we already hit all of them, or is there still some coming up? Yeah, I'm going to keep going to make sure I cover them. Mitochondrial DNA, you inherit it from your mother, males and females, everybody. You can trace maternal lines through mitochondrial DNA. This is very simple. The egg has all the mitochondria. So little mitochondria, right? Little powerhouses of the cell. So the egg, that's not very well. There we go. The egg, so there's its nucleus. All these are mitochondria. The sperm does have mitochondria in it, but here's the deal. The sperm, when it fuses with the egg, just the genetic information from the sperm goes into the egg, not the mitochondria. Mitochondria self-reproduce. That's really maybe odd to hear, but mitochondria reproduce themselves. They actually have a little bit of DNA inside of them, and they actually make themselves. And so all of your mitochondria that are in your body came from your mother because the egg was what had the mitochondria. Okay, it actually is like a small chance you can inherit a little bit from your father, but very, very little chance. Questions? And then the Y chromosome is actually how you trace, of course, paternal lineage, is the Y chromosomes passed on from male to male to male, and so you can actually trace it that way. All right, chromosomal damage. These are all kinds of different ways that chromosomes can be damaged. I don't think you need to memorize those specific ones. I'm hoping not. I'm looking for learning objectives. I want to make sure I cover them for you. Translocation's wonderful. Now this starts to, boy, I'm thinking you have some stuff on this. Yeah, aneuploidy. Aneuploidy is a fancy way of saying you don't have the right number of chromosomes, one way or another. And that is a big deal. There's only a few diseases in which you can actually survive and have those, and that's trisomy 21, which we're actually seeing right here, Down syndrome, trisomy 21. Trisomy 18 and trisomy 15 are the other two, but they're more rare. Trisomy 21 is by far the most common. 18 and 15 also occur a little bit, and those are 18 is what, Petai syndrome, and Edwards syndrome, I believe, is 15. So be able to identify the trisomies. There is this is where you only have one chromosome, Turner syndrome. That's when someone has a genetic condition of XO. They literally don't have an X and an X, or an X and a Y. They have an X and an O. Female phenotype, but frequently a sterile individual doesn't have the ability to reproduce. Turner syndrome. So here's Down syndrome. Describe the chromosome disorder that causes Down syndrome. Relatively common, 1 in 691 births. Has to do with having 3, that's that trisomy, 3 chromosome 21s. Any questions about anything you've ever wondered in that area? Okay. Explain and provide examples of multifactorial inheritance. Oh yes, do know this one. Make sure you review this just so you're really sharp on all these parts. This is where you have something caused by multiple genes and potentially environmental factors. Good examples are that cleft lip or palate. You actually see that right over here. This is where it's not 100% genetic. It's not 100% environmental. It's actually a combination of the two that results in the actual disease. Yeah. There's other ones here. Congenital hip dislocation and heart disease, as well as clubfoot, also fall into those categories. And then teratogens. Teratogens are any sort of environmental factor that can cause birth defects as a result of exposing the fetus or the mother to these things. And what are they? All kinds of things, right? From radiation, so that's why pregnant women, they're not subjected to the same amount of radiation exposure that others might be. Lots of drugs and chemicals such as alcohol. I'm sure all of you have heard of fetal alcohol syndrome that can result from that. Anticoagulants, warfarin, can actually cause birth defects if they're used during pregnancy. Anticonvulsant, cancer drugs, all kinds of things can actually do that. As well as, know your categories especially. So drugs and chemical substances, radiation, infectious things, certain viruses and diseases can actually result in pretty substantial effects on the fetus, as well as some non-viral factors. These are just what do you call it? Wow, I'm so bad with names whenever I'm not frequently in the classroom. Single cellular organisms, protists, what's the right term? I'm thinking of protozoa. Yeah, these are all protozoa of different types. Protozoa, so single cell, but eukaryotic cellular organisms that are pathogenic. Toxoplasmosis, I'm sure you've heard, don't change the cat litter box whenever you're pregnant, because cats carry toxoplasmosis, and you can get that and cause major problems with the fetus. Period of vulnerability. This is where, here's the key thing, it's actually really early on that there are the major things developing, and then it kind of slows down. So the process of organogenesis, which is the production of the different organs, happens during the first few weeks. Why am I so like, oh my? Well, often a female may not recognize that she's pregnant during that phase of development and may not then avoid various teratogens, which actually are most potent often during this very rapid phase of development, really early in fetal development. And then our last one here. Yep, so Mrs. K is a diabetic, having trouble keeping her blood glucose constant during pregnancy. She works in a dry cleaner store next to a gas station, drinks heavily. She's diagnosed with toxoplasmosis. What kind of stresses is her fetus experiencing? Have mercy. All kinds of stresses, all those things can be teratogens. So there you go. Actually, especially these last two there. Real quick, if you want to see it, I will show you 23andMe before you run. It's kind of a fun little thing. I'll show you what that looks like. And then we'll go from there. All right. So on the screen here, just in case you're curious, this is just like a little report that you can get. This is my sister Debbie. So between me and Debbie, we share about 45.5% of our DNA. Here's chromosome 1. It's showing what percent of chromosome 1 we're sharing, the half identical and then the completely identical. If I go back, there were kinds of reports that you can get or all kinds of interesting things. I'll actually show you this. This is about our little Ethan. In fact, I'll show you his DNA relatives. This is probably the more interesting way to
We're
look.
nerds, and we
Yes.
have genetic testing done on our children.
Oh, that's true. Actually, it's kind of like bad in some ways, isn't it? Yeah. They're like 4 and 6, so anyhow, they weren't able to sign consent. Mm-hmm.
Between me and she's free, but the question is, what combination from their grandparents did they get?
Yep. And in fact, let me actually switch over to Ethan so you can actually see that. There we go. So the DNA relatives thing shows you just what percent of your DNA do you share with anybody in the database, right? And you can volunteer to get into the database or not. So of course, with me, he shares, although it says 47.6, Ellen, no, I'm kidding. It actually is 50% because they include the X chromosome, and that looks like extra, so that's all that is. But here's Emma, his sister. He shares 44%, but then here's the interesting thing. Here's Grandma and Grandpa. So his grandpa on Ellen's side, 30% roughly. On my side, only 22%. With his grandma on my side, it's about 25%. Grandma on Ellen's side, about 22%. So as you can see, as it goes on, you actually have different percentages all the way down. Here's a second cousin, if you want to see what a second cousin looks like, and then I'll let you run. So, beware, there are risks with doing this, and your major risk is, if you voluntarily do this, you put your DNA information out there, it's kind of like how you have Facebook or Google or anything else, and they know all kinds of stuff about you, and you're signing this long form, and then, years down the road, someone's in front of a congressional hearing saying, why are you abusing their information? There are risks in putting your information out there. Be aware of those risks. I'm not pushing you to do that. But anyhow, there you go. Have a great day, everybody. It's good spending time with you guys.
Thank you.
Yes.